I'll introduce Rob Balshaw from CFI Data Sciences, and he's going to give us a very brief overview of what they do, how to engage with them, and then Rob, I'll get you to hand off to Megan immediately after so she can introduce our speaker.
Hello, I'm starting to recognize quite a few people. I've only been in University of Manitoba for a couple of years now, and really enjoyed it, recognizing many of the faces here. And I wanted to, well, I heard that the masterclass was happening, and I thought I was going to just butt my nose in and say, could we please have a couple minutes to say hello, let you know about the Georgian Fayet Center for Healthcare Innovation, particularly the data science platform, and the group that I'm involved with in the data science platform, in particular the Biostats consulting group. But we also have a representative from one of our other groups. We've got Dr. Pingzhao Hu, who is our bioinformatics specialist. And we have a third group, the Clinical Research Data Group, and some specialists in that group weren't able to make it. So wanted to say hello, tell you a little bit about the center. Okay, it's the Manitoba SPORE unit. That's the Strategy for Patient-Oriented Research that CIHR funded a number of years ago. We're here to help with you folks. You guys are researchers in the biomedical community, and you are looking at patients. And that's what the Center for Healthcare Innovation is about, why we're here. Comprises, excellent. I think I'm being offered the opportunity to update. No, I can search. Anyone tell me how to go backwards with one hand? Left. Left. There, thank you. All right. The CHI has 7 platforms. I cannot speak knowledgeably about any of them except for the group that I'm with and the knowledge synthesis group. We have one of our faculty members is actually co-appointed in the 2 groups. And her specialty is meta-analyses. I'll say a bit more about that in just a second. This is the key slide. We're here to help. If you have data, if you will be collecting or gathering or working with data, if you are even starting to think about data, please give us a call. That's why we're there. We do right from grant proposals or thesis brainstorming all the way through to preparation of manuscripts or assistance with interpretation of the methods that you've been struggling with in your thesis. That's why we're there. We do coaching, we do collaboration, we do consulting of all types. We can help with study design and sample size calculations, methods, analyses, statistical programming, and the interpretation of the methods that you've been working with. We also have, this is where, in particular, the REDCap and Data Management Services Group, our Clinical Research Data Group, I always get that wrong. The Clinical Research Data Group is there to help you when you need to store your data and we need to keep high quality data organized. Okay, so if you are struggling with your data management plan, this is a group you might want to speak with. Okay, and if you are a student or trainee, you're entitled to up to 5 hours of fully subsidized support from us, largely in the form of coaching. Please don't drop your data off on Friday and ask for the presentation to be ready on Monday, because it's not going to happen. But if you're struggling with whether it's a logistic regression or maybe a discriminant analysis, or this is longitudinal data and that's just not what I'm comfortable with, that's exactly where we're going to be able to help you. We also, one thing I didn't manage to get in here is training. We also offer a number of training courses, right from introduction to R, from I can barely spell it, all the way up to our specialists in latent class modeling, latent trajectory modeling, and beyond. And if you are working on a grant, we would absolutely love to talk to you. We'll help you with the budgeting for statistical support. And then that means that there's going to be statistical support in your budget, which means we want you to come back and we'd love to work with you. It is a cost recovery service, but it's, we try to keep things moving and as affordable as we can keep it. All right, just to give a sense, we start with, I mean, we do surveys, we do clinical trials, we do everything in between. Patient-reported outcomes with the SPORE program in particular gives us a bit of a focus. We've got experts who are particularly good with working with the MCHP databases. I have a bit of a smattering of the omics that I can never seem to get away from and love to work with it, so please come talk to us about that. Disease areas, conditions all over the place, anesthesia, transplantation, diabetes. Many of these will be familiar to you folks. Okay, and methods, this is just showing off all sorts of expensive words in here: causal inference, structural equations, latent variable modeling. Please do, if any of this is of interest to you, or you'd like to learn a little bit more, or talk to people who can maybe help you with these, give us a call. So if it isn't clear, call us early, call us often, let us help. That's really why we're there. You folks, many of you are already into the $1 million+ grants, and that's $1 million grants, and that's wonderful. We'd really like to talk to you, make sure we get involved in those. And the rest of you who are just starting your careers or are reshaping your career, statisticians aren't necessarily evil, and we would love to hear about your research. That's why we do our work. Okay, so thank you for the opportunity to address the group, and I think I'm supposed to hand this over to Megan.
Thanks for that. I'm excited to start working with the group. Let's make sure that this is okay. I'm very thrilled. I'm here to introduce our speaker for today, Dr. Kaitlin Wade, who's going to talk about Mendelian randomization, which is one of these amazing buzzwords that's becoming more and more popular. People are hearing about MR, or learning more and more about it. But I was asked to come and talk about why MR is important for DOHaD. And I think that the real sort of key to this is it gets at this idea of causal inference. A lot of traditional methods in epidemiology where we're connecting early life environments to later life health outcomes are associative. So we can say these things are connected, but we can't necessarily say what causes what. Co-causality is an issue, population structures is an issue. And so Mendelian randomization is one method where you can actually start inferring causal relationships between these associative variables. So Dr. Kaitlin Wade is here. We're extremely lucky to have her, especially now because we're all missing DOHaD International. We're sad about that, but we're very, very happy to have her here with us today. And Dr. Wade has got an amazing pedigree in the MR field. She did some of her training with Dr. George Davies-Smith and works at that MRC epidemiology group in Bristol that has really pioneered the field of Mendelian randomization. She was involved in the MR base resource, so for people who are new to MR, I do recommend checking that out. It's sort of a curated online platform for learning about MR, and it will actually do some MR for you once you learn how to use it. And to show her research is on using MR to infer causal relationships in a lot of different areas. Some of the ones that are particularly relevant for us in the DOHaD group is she's done a lot of really beautiful work on obesity and diabetes. She's got more than 35 publications, some of them really, really amazing, really foundational work looking at causal inference in a lot of these areas. She's an early career fellow at the Elizabeth Blackwell Institute at the MRC Integrated Epidemiology Group. And like I said, that group is really, really powerful in this field. They are doing some of the best work in the world. So we're very lucky to have her, and I'm excited to hear her speak. Thanks.
Great. Hi, everyone. Oh. Great, hi. I'm absolutely honoured to be here. This is really exciting for me. I've never been invited to come and speak before, so this is a new stage in my career that I'm very excited about. So yeah, as Megan said, I work at the University of Bristol, so specifically, as you can see on the bottom right-hand side, the Medical Research Council funded unit that has really, as Megan said, pioneered Mendelian randomization as a method to try and overcome lots of these limitations of observational epidemiology. So I suppose one of the things I wanted to talk about today was what Mendelian randomization is. mainly just to kind of make sure that people, if you really want to use it, then you know kind of some of the assumptions behind it, the limitations that it has as a study design, and where to go from there. So I was asked to give some learning objectives, like a teaching experience. So one things I'll do are explain Mendelian randomization and its assumptions and where it fits in observational epidemiology, describe the strengths and limitations it has as a particular study design, and then I'm just going to illustrate a couple of examples that it's been used to improve causality in lots of different contexts, some of which I've been involved in, and then I might just finish on just talking about some of the work that I'm doing at the moment and where it kind of fits in terms of Mendelian randomization and that kind of method. So just to kind of put a little bit of an outline on this talk, I've got a little bit about epidemiology and where Mendelian randomization fits. And to do that, I'm going to discuss the framework of MR and its assumptions, the methodologies that you can actually use to create causal effect estimates, and again, just talk about some of the limitations of that study. And then just wanted to talk a little bit about some recent methodological developments that we're really excited about, one of which is the MR-based platform that Megan introduced. And then just a couple of examples of where I've been using it, particularly in the field of understanding the links between the gut microbiome and various health outcomes. So I'm sure everyone here is aware what epidemiology is, but just to kind of really bring it back to what we're interested in, is looking at specifically the distribution or determinants of particular health outcomes in specific populations. We could be interested in lots of different things like cardiovascular disease, all the way up to kind of mental health illnesses, and we really want to know what causes or prevents these particular things, mainly so that we can prevent or treat them in the population. And as Megan said, it's kind of difficult to know what causes particular things when we're looking at associations in observational studies. So what we tend to do is look at the relationship between a particular exposure. So we've got X denoting the exposure, and then our particular outcome. So I do a lot of work with adiposity and obesity. So for example, if we're interested in the relationship between BMI, body mass index, and cardiovascular disease, we can do this kind of thing with lots of cohort studies, cross-sectional studies, that kind of thing, but there are some problems with this kind of framework. So there can be confounding factors, so things that cause or are at least associated with body mass index, in this case, and cardiovascular disease. There can be reverse causation, so it's completely plausible that if you're looking at, say, an older population, people who have cardiovascular disease and those more comorbidities that come with that, they may lose weight as a result of those comorbidities. So you may actually see an inverse association between body mass index and cardiovascular disease. And there are lots of different biases that sometimes we can't really account for in these different types of studies. So recall bias or reporting bias, for example, it's very plausible that people with a very high body mass index may not report that as accurately as people on the rest of the scale. So if these things aren't really taken into account, conventional epidemiology can get the wrong answer. And we can do this kind of analysis in what we used to sort of think of as a hierarchy of causal evidence, everything from ecological studies at the bottom to what we kind of witness as the best type of causal evidence at the top with a randomized control trial. So I'm not going to go really into much of these particularly, but just very briefly for those of you who are interested or don't know, cross-sectional studies, we look at one time point, so it's really difficult to decide which particular phenotype came first in that relationship. Case control studies, in the example of body mass index and cardiovascular disease, we take people who already have cardiovascular disease and look back into their history and see whether there were any differences in body mass index. Cohort studies, which is again something that we use quite a lot in Bristol. For example, I'm kind of here representing the AUSPAC study for the children of the 90s. And what we do there is recruit people from a specific time point in the AUSPAC cohort. It's from birth. And we follow those people up over time, and we can see kind of temporal relationships between particular health outcomes and the exposures that should have preceded those. And then with randomized control trials, what we like to do is randomize people to a particular drug or intervention, and then we can, by that random assignment, literally, but sometimes by flipping a coin or rolling a dice, we can equally distribute any of those confounding factors like diet, education, socioeconomic position between those two groups. So any differences we see in the outcome between the groups should have been due to that randomization effect on the exposure. And that's kind of why we think of it as kind of the best causal evidence. But there's actually kind of a movement now to sort of just, rather than there being a hierarchy where one is better than the other, we're trying to move towards this idea of triangulation, so understanding the limitations with each of these different study designs, using them and comparing the findings just by understanding what those limitations are. And I'll talk a little bit about that on Thursday if anyone's coming to the conference at CHRD. So as I said, randomized control trials are usually what we think of as the kind of gold standard, but sometimes this can bring results that maybe weren't anticipated. So one thing that I'll kind of keep as an example dotted throughout this talk is going to be the example of selenium and prostate cancer. So about 10 years ago now, there was lots of observational evidence suggesting that selenium, which is a kind of blood metabolite, which you can obtain through various supplements or dietary, specific dietary intake. There was lots of observational evidence suggesting that an increased selenium supplementation reduced the risk of prostate cancer quite substantially by about 25% with almost minimal supplementation. And that then spurred the idea of generating a randomized control trial to test causality in this relationship. So this led to the initiation of the Selenium and Vitamin E Cancer Prevention Trial, or SELECT. And it was set up, I think it was supposed to be set up for 12 years, and the idea was to test whether or not people taking selenium supplementation or non-selenium supplementation had any differences in prostate cancer risk. So this is just an excerpt of the abstract from that first paper, trial paper. And actually what they showed, so first of all, it was supposed to run for 12 years. It was actually stopped after 7, and it cost about $114 million, something like that. And actually what they showed was no evidence really to support at least an inverse association between selenium supplementation and prostate cancer. If anything, the hazard ratio actually showed an increased risk of prostate cancer and advanced prostate cancer. And there was some off-target effects as well. One of the most publicized at the time was type 2 diabetes. So obviously it was stopped. There were some detrimental effects. And this wasn't particularly predicted based on that observational evidence. It's leading to the fact that that observational evidence may have been confounded in some way. So we see, this is kind of where Mendelian randomization fits in, as the idea is that we want to try and predict what may happen in a trial or understand causality between relationships when actually we don't want to do the trial, or even when we're unable to do a trial. For example, Mendelian randomization has been used quite a lot where trials would be unethical in that situation, so probably couldn't randomize people to smoking a lot of, well, cigarettes or injecting heroin, so in that case we could maybe do that in a Mendelian randomization analysis. So that's kind of the motivation of MR. So I'm going to talk about what MR is and just describe it in a couple of different ways in the hope that one of them may stick. So if we kind of think about genetic variation, so Mendelian randomization is inherently using genetic variation. And this is just a couple of statistics from a couple of years back now, trying to just emphasize the fact that there is a huge amount of genetic variation just in the population at any one time. So if there's about a mutation in about 1 in 110 million base pairs, roughly, that means that every single newborn has about 60 or more new mutations. And if there was about, I think now about 140 million births in the last year, that means that every single base pair in the whole genome has been mutated at one time. And that type of thing doesn't account for the things that we inherit as well. So there's a huge amount of genetic variation that importantly is randomly assigned at conception. So this is essentially equivalent to a natural experiment where we have proxies or a particular randomization event that has happened that we can then use to our advantage. And one thing that we actually know now quite a lot of, and this is actually an outdated slide, is that we know now a lot based on genome-wide association studies what particular variation at the genome level does to various health outcomes or traits. For example, things from cardiovascular disease, immune and nervous system diseases, cancer, even responses, how people respond to drugs. So we know a lot now about what genetic variation does, and we can use that in our advantage with Mendelian randomization. So Mendelian kind of comes from the idea of Mendel and his laws. So the main law that we try and, or the kind of corollary, or whatever you want to call it, was the idea that each pair of characteristics that we have is inherited independently from one another and at random. So if we compare people or groups of people in the population based on a particular genotype or genetic variation, then they can be used for different proxies or biomarkers for otherwise confounded modifiable risk factors. So similar to a randomized control trial, they shouldn't be associated with kind of confounders. So those confounding factors of our exposure outcome association should be equally distributed between groups defined specifically by genotypic variation, meaning that we can kind of use it to infer some causal inference. So I'm going to do a lot of these diagrams over this talk. So basically any arrow essentially means an association or potential causal association. So again, with this same framework, we're interested in the relationship between an exposure and an outcome. Plausibly, there could be some confounding factors or bias or reverse causation. And what we do with Mendelian randomization is use a genetic variant or variants associated with our particular exposure as an instrument. So the word instrumental variable comes from econometrics, which is where this method essentially arises from. So you kind of you see the word instrument or instrumental variable or genetic variant essentially used interchangeably in the literature. So we can use a genetic variant as an instrument, in this case, to proxy for our exposure of interest. Now, there's a whole bunch of assumptions that Mendelian randomization relies on, but there's essentially three that people tend to consistently talk about and also try and test in their studies. So one is that we want to make sure that our particular genetic variant is associated with our genetic variant, otherwise it wouldn't be a very good proxy. That's sometimes called the relevance criteria. We want to also make sure that our genetic variant is independent of those confounding factors of the exposure outcome association. That's sometimes called the independence criteria. And then we also want to make sure that there's no independent pathway linking the genetic variant that should be associated with our exposure and our outcome of interest. And that's sometimes called the exclusion restriction criteria. It's sometimes called pleiotropy. Simply that there should only be a relationship between the genetic variant and the outcome through its effect on the exposure, which is very similar to what you think of with a trial. Say you've got a trial of statins. There will be a relationship between statins and your outcome of, say, coronary heart disease, but the assumption is that that's only through its effect on LDL cholesterol, for example. So this is kind of the diagram or directed acyclic graph or whatever illustration of what Mendelian randomization is. And we can use this because of two main kind of random events. So first of all, we have random segregation of particular genetic variants or alleles at conception. And then we have a different or a random distribution of genetic variation at the population level, much like a trial, rather than literally giving someone or randomly assigning someone an intervention. We have a random assigned population of genetic variation, which we can use with MR. So what we essentially do is compare groups of individuals defined by a particular genotype that should only differ with respect to that genetic variant or its effect on an exposure. And Megan actually mentioned kind of population structure and stratification earlier. So with the control of population structure by using, say, homogeneous populations, by the way that they've inherited ancestrally their genetic variation, there shouldn't be any confounding by things like behavioral or socioeconomic or physiological factors that would otherwise confound that exposure outcome association. So this is just to kind of reiterate that point. So we have the three main limitations of observational epidemiology. And the way that MR gets over these things is by, first of all, independent assortment, so that random distribution of alleles at conception, which gets over this confounding idea. The fact that our genetic variation is laid down at conception, there should plausibly be no biological reason why the outcome can affect our genetic variation. And there shouldn't be much measurement error as a proxy for different types of bias that we have, because we're very good now at measuring genetic variation, either with genotyping or sequencing, for example. So this is largely reduced as well. And just to kind of reiterate the point of its analogousness to randomized control trials is kind of similar to the illustration that I showed before. So on the right, we have the randomized control trial, again, randomly assigning people to either an experimental intervention or a drug. Any differences we see in the outcome should be due to the effect of that randomization on our exposure of interest. Similarly, in Mendelian randomization, we have random segregation of alleles at conception. So we can look at people who differ with respect to their particular allele structure at either one particular locus or multiple loci. So any differences that we see in our outcome of interest should be due to that random association of our genetic variants on our exposure of interest. And just to kind of come back to this sort of confounder equal distribution, there was a really nice paper recently, it was about 10 years ago now, that looked at the relationship between various confounding factors and genetic variation. So this is a paper by Debbie Lawler and George Davey Smith. And what they looked at was the pairwise correlations or associations between genetic factors and non-genetic factors. So we have 96 non-genetic factors here, so things like education, smoking, socioeconomic position, alcohol, intake, that kind of thing. So they had just over 4,500 possible pairwise associations between those 96. And then essentially the last three columns are what would go into, say, a chi-squared test. So you have, if you set a p-value of 0.01, literally meaning that you expect 1% of those to be significant, you would expect 10% of those, so about 45, to be significant. But actually what you see is 45 times more relationships than what you would expect by chance. And that makes complete sense because people who are more educated have a higher socioeconomic position, people who drink more likely to smoke. So everything in our observational world is very associated. When you look at genetic variation, the opposite is the case. So they did have a 250, but again, if you expect 1% of those to be significant, you don't really see that much more different than what you would expect by chance, suggesting that genetic variation is much less correlated than observational traits. And actually, when you combine the two together, again, you don't see anything much more than you would expect by chance. So that genetic inheritance is randomly assigned and not associated with those particular non-genetic traits. And then one thing I wanted to talk about, so talked about the kind of framework of MR, talked about its assumptions, and I wanted just to give a couple of examples that actually demonstrate a lot of the graphical things that you see in some of the literature, so hopefully that will make a little bit more sense. So here what I'm showing is an example of the relationship between a marker of inflammation, so CRP, C-reactive protein, and triglycerides. So this is our analysis of observational individual level data from a cohort, let's say it was ALSPAC, and we have a nice positive relationship between log CRP on the x-axis and triglycerides on the y-axis. Each of the dots represents an individual, so nice positive relationship there. So we may conclude that, okay, well maybe CRP could be used to, or reducing CRP in the population could be used to reduce triglycerides. But what can we do with Mendelian randomization? So the colors here represent the genotypes of particularly 3 different CRP genetic variants, so actually within the CRP gene. You don't really need to know what the colors are representing, but essentially what we're doing now is stratifying our individuals based on 3 different CRP genotypes. I mean, we can't really tell much here, but what we can do is say look at the means of CRP, the actual protein, by those CRP genetic variants. Again, it's explaining, so one thing that I'll come back to is variation explained in our exposure. So here, the scale observationally has reduced much, much smaller when looking at the means by CRP. So if we zoom in a little bit, the colors have gone, but essentially the idea is that we do, if we look at the x-axis, we can see variation in the actual phenotype of CRP protein by genetic variation. It's not horizontal, it's not vertical, so there's a nice variation explained in CRP by the genetic variants in the CRP gene. So what we can do by this is add some error, much like you would do in any kind of regression analysis, add a fitted line, and then add our confidence intervals. And then if we zoom out again, what essentially we've done is done in a Mendelian randomization analysis, because we've looked at the levels of triglyceride variation by the CRP genotype. So the blue line here is the observational relationship between CRP and triglycerides. And then the green line essentially is completely discordant with that. If anything, it's flat or potentially negative. So what we've shown is that actually Mendelian randomization doesn't support this causal effect of CRP and triglycerides. And there's just a couple of things to note with this kind of inference as well. So first of all, the variance explained by the CRP gene is incredibly small. So we've got a huge observed level of CRP, but when we look at the means of that in the CRP gene, it's very, very tiny. And that makes sense, especially when we're looking at common genetic variation. So by the fact that it is common in the population, the effect sizes that they have on different traits are very, very minimal. Otherwise, it would have been selected out by natural selection, which is why lots of rare genetic variation has massive comparable effects. So the variance explained usually in our particular exposure of interest is usually fairly small, which means we need massive sample sizes in Mendelian randomization. And also the extrapolation from that tiny proportion of variance explained means that our confidence intervals become very, very wide at the end of those tails, because we simply just don't observe any CRP at those tails. So we need larger sample sizes to try and account for the fact that that standard error may be larger in our MR studies. So this is just a kind of numerical summary of that plot. So the linear regression showed some increased relationship between CRP and triglycerides. But again, the instrumental variable or Mendelian randomization approach showed null results, if anything, potentially negative. And this p-value at the bottom is just testing whether there is a difference between those two estimates, which there seems to be, which you can see by the confidence intervals. So yeah, we might then conclude that whilst observationally we saw a relationship between CRP and triglycerides, this may suggest that actually CRP lowering drugs may not be a good target for reducing triglyceride levels in the population. And then a couple of just other things. So when we think of Mendelian randomization, what we're looking at is genetic variation that has laid down at conception. So it's a lifelong effect of increased CRP. So what we tend to see is larger effect sizes when it's consistent with our observational effect, but actually what we're tending to say is that it's a lifelong effect of an increased exposure level. And actually, if you compare that to, say, a trial, the drug trial only lasts, in the SELECT trial, for example, only 7 years, and that's only a 7-year intervention. So that's just a slight comparison to what we're seeing between MR and randomized control trials. So that's Mendelian randomization. I was going to say, has anyone got any questions before I move on? Yes.
So you selected this method.
Yes.
Let's go back to that.
This one? That one. So I think there were 3 SNPs. They're associated with CRP. And usually they're within, so in this case, these were within the CRP gene. So biologically, it's very plausible that they can only have an effect on the protein directly. Yeah? Yeah, that's a really great question. So I'll come on to that, actually. You're about to preempt what I was going on to. But essentially what we do is we select SNPs that are robustly related with our exposure, usually derived from genome-wide association studies, or if you don't have one available, say a candidate gene study where you know that there's a relationship, potentially biological, with your particular phenotype. There's a movement towards including more and more and more SNPs in your, or genetic variants in your MR analyses. But actually, with doing that, you're introducing possible pleiotropic associations, so more likely that you've got an independent pathway between your variant and your outcome. And I'll go on to some of the methods that can be used to sort of test those pleiotropic effects. But usually what we say is that, so for example, there are tens of SNPs that have now been related to CRP in genome-wide association studies, but still consistently, most of the MR studies on CRP use the same ones in the CRP gene because there's better biological knowledge of what they're doing functionally. So yeah, that's kind of how we do it. I think that was just the effect size, so this kind of, yeah, the effect size of the genotype or allele on CRP. This is kind of more of a theoretical example. Yeah. That's all right. Does anyone got any other questions? Oh, yeah, that's a good idea. Yes. Yes, definitely. So people tend to use single SNPs individually, but there have been studies and there are plausibility, there's certainly ways in which you can combine the effects of different SNPs as well. Sorry? I think they were all just used independently for that example, yeah, but there have been studies. You can combine them to actually physically look at their kind of separate and combined effects. Most people tend to use them individually and essentially meta-analyze them at the end, which again assumes that they're essentially working independently, but you can totally look at interactions between them or the effects of the combined SNP effects, yeah. Any other questions before I move on? Cool, okay. So I've talked about what MR is, how we can kind of infer causality from it. I just wanted to go through some of the methods that can be used to derive causal effect estimates in that Mendelian randomization framework. This is a very short list compared to what is available, so I'm going to only go through a couple of them, but if anyone's interested in any of these particularly, I'm literally here all week, so I can answer any questions you want. So very briefly, I'll go through individually, but I'm going to mainly talk about the second and the third one here. So the SNP disease association is essentially when you have a particular data set that doesn't have the exposure measured. So what you're genuinely looking at is the relationship between your SNP and your outcome, which might seem counterintuitive because we have that main assumption that there is no independent link between your SNP and your outcome. But if you see a relationship in that regression or whatever analysis tool you want to use, that's assuming that the only reason that exists is through the exposure. So it doesn't allow you to test whether it is independent of your exposure because you don't have it measured, but it allows you to infer that maybe there would be a causal effect. So you can go on to various cohorts that do have everything measured and do it formally. And the way that we do it formally, again, this is only two of the many examples of the ways that you can generate causal effect estimates using individual level data, is the Wald ratio and two-stage least square approach, which I'll go into in a second. And this is when we're using, again, individual level data. So this is sometimes called one sample Mendelian randomization, which some of you may have heard in the literature, is essentially just using an individual study that has the exposure, the outcome, and the genotypes that you want measured in one sample. Two sample Mendelian randomization very much does what it says on the tin. You have two independent samples, and usually this is sometimes called summary level data Mendelian randomization because you don't need individual cohorts to do this analysis. And this will make a little bit more sense when I explain what the world ratio is. But essentially, you're using summary data, so beta coefficients, standard errors, p-values from an existing cohort or regression that's existed, that's come before. And I'll talk about those two specifically. And then the last three are just essentially extensions and can be used with both individual level data or summary level data. So bidirectional MR is when you genuinely just look at both directions for your exposure and your outcome, and the reverse. Two-step MR is something that may be relevant here, is used to understand mechanisms or mediators. So you literally look at the relationship between your exposure and a mediator, and then look at the relationship between your mediator and your outcome. So testing whether you think there could be a causal pathway between your exposure and your outcome through a third variable. And that was originally proposed using methylation data with George and Caroline Relton at the University of Bristol. And then gene-environment interactions are a really kind of new way of thinking in terms of if you have a particular genetic variant that you think only works in a particular environment. So for example, one of the best characterized SNPs associated with smoking is not associated with whether or not someone smokes. It's actually related to heaviness of smoking. So in that case, you shouldn't see a relationship between that SNP in non-smokers, that kind of thing. So it's kind of a nice test of whether you think that there's an independent effect of your SNP on your outcome in a population sample that it shouldn't have an effect. So I'm just going to go through these two that are starred. So the world ratio is something that we can use and was first initially proposed with individual level data. So when I started out doing this, two-sample MR didn't exist. You had to have an individual cohort data set that had everything measured. So in this case, your causal effect estimate can be derived simply by the division of the regression coefficient between your instrument and your outcome, divided or scaled by the beta coefficient linking your instrument and your exposure. That is as simple as a causal effect estimate, and that's what the world ratio does. So both betas are regression coefficients, either through linear or logistic regression or what other regression coefficient or analysis you want to do, and that is your causal effect estimate. You can generate the standard errors in whichever way you want, but this would be your causal effect estimate. Similar to this, and this actually now requires individual level data, so something called a two-stage least squares approach is essentially the same thing in terms of generating an estimate that reflects a per unit increase in your outcome, per unit effect of your outcome per unit increase in your exposure. It's a similar way. So what you do is you regress your exposure on your genetic variant. You take the predicted values from that regression, so essentially the genetically predicted levels of the exposure, and then you regress the outcome on that. And you can generate the standard errors again in whatever way you want, but essentially both the world ratio and the two-stage least squares, the actual effect estimate you get from that is the causal effect of your exposure on your outcome. So those are both using individual level data, and then literally what must have been 6 years ago now, there's this new evolving way of doing Mendelian randomization where you don't even need individual level data anymore. So you don't need Ausbach, you don't need child, you just need access to GWASs that exist. So if we think about the world ratio, so this thing here, these are just beta coefficients. So there's no real reason why these can't come from existing beta coefficients that have already been used. So what two-sample Mendelian randomization does is use one sample, which is usually a GWAS of your exposure, where you have identified SNPs or genetic variants that are associated with your exposure of interest. You get the beta coefficients from that one sample. And then the second sample, which is usually, again, a GWAS of your outcome, you look those same SNPs up in that GWAS of the outcome, and say it's a binary outcome, so you get, say, a log odds ratio, of the relationship between those same SNPs that drive variation in your exposure and look at those in relation to your outcome. And then your causal effect estimate is essentially just a Wald ratio. So you have the log odds ratio divided by the beta coefficient. And then if you have multiple SNPs, you can do that individually and meta-analyze them, and then you have your causal effect estimate through those genetically predicted exposure variation. And actually, this method is something that's being used a lot more in the literature, mainly because the advent of genome-wide association studies is getting so much bigger. You know, there's a GWAS that comes out pretty much every other day. And actually, this is really useful for when, in, say, a DOHaD type field, where it's very difficult to get large enough sample sizes for Mendelian randomization analysis that also has very early life, if not prenatal exposure measures, and very late life in the offspring. So there are child study, ALSPAC, that has got really nice comprehensive data on those, but it's very difficult for every question to be answered with a one sample individual level data. So this is quite nice for those questions. So I've talked very briefly about the framework, some of the methods that we can use, and I just wanted to talk about, first of all, how sensitive Mendelian randomization is to those three main assumptions, and then go on to some of the limitations that we need to think about when we're applying this method in real life. So these were the three assumptions again. So we have the relevance assumption, which is the effect of essentially that we need some association between genetic variation and our exposure. We want no association between our variants and confounders, and we don't want any independent pathway linking the genetic variants and our outcome. So I'm just going to go through briefly why these need to be met and how we can try and test that they are met. So first of all, this first assumption. So I'm sure most of you have seen this graph, but essentially we can use genome-wide association studies. Most of the time they showed Manhattan plots. So you have the chromosomes along the x-axis, the log base 10 p-value on the y, and each dot represents a genetic variant in the genome. And essentially the height of that dot means that we think that it does something to our particular trait that we're testing. And these are coming out all of the time, so we now know quite a lot about what genetic variation does in terms of traits or diseases at a population level. And just as an example, so again, I do come from a place that does a lot of work on body mass index and adiposity. So just as an example, this is the landscape of genome-wide association studies for body mass index. So in 2008, there was a study conducted by Ruth Luce at Mount Sinai in New York. They found 2 hits kind of robustly related to body mass index. So that was, 11 years ago now. In the same year, they increased the sample size again and found 8 signals. 2010 increased the sample size to about 240,000 individuals and found 32 genetic variants across the genome, which kind of makes sense. Body mass index is fairly polygenic. Across the genome associated with BMI, this little FTO guy seems to keep coming up in all of the genome-wide association studies. The most commonly used one is the result from what Adam Locke did in 2015, which is with the GIANT consortium, which has now been kind of fully generated with 339,000 individuals, and they found 97 genetic variants, where the FTO variant is now not on the graph. So now the p-value is, I think, 5 times 10 to the minus 150. So quite a strong relationship between FTO and body mass index. And most recently, we have obviously the advent of UK Biobank, which is an amazing resource in the UK. And what they did was combine this study with UK Biobank and found in about 682 individuals, 941 genetic variants associated with body mass index. And the p-value, I'm not sure if you can see it, is now, the top hit is of 1 times 10 to the minus 2,000. So huge association between FTO and a lot of other signals, but mainly FTO, which is and body mass index. So now, we can use all of these in a Mendelian randomization analysis, which is brilliant. We have more variation in the genome, which explains more variance in our BMI variable. So that increases our power, increases our ability to detect an association between BMI and whatever our outcome is in an MR study. So these kind of landscapes are changing again and again and again to help us with these types of MR studies. So with genome-wide association studies, we can identify these variants. They're usually, because of the number of tests that they're doing across the genome, everything from a couple of 100,000 up to now kind of 800,000 in the genome, they tend to have the possibility of identifying variants due to false positive results. So they tend to have a very strict p-value threshold, so usually about 5 times 10 to the minus 8. And that gives us an idea about how strongly and potentially reliably our genetic variation is associated with our exposure. But ideally, we want something that has been replicated in multiple studies. Again, just giving us confidence that we think that it is not due to chance that our variant has been associated with the exposure. So that kind of first assumption is testable. We can look at variation and we can find it that has been related to our particular exposure. The last two are slightly more difficult. So the first is that we want to make sure that the variant is not associated with confounding factors. And obviously, we can do that if we have individual level data. We have things measured, like if we're interested in the association between BMI and cardiovascular disease, we can physically check the association between our genetic variation and things like smoking, diet, physical activity, that kind of thing. But we obviously can't test for anything that we haven't measured or observed in our population, and we can really only rely on the fact that what we have measured is a good proxy for the kind of comprehensive possible variables that could confound our relationship. So we have to rely on really good knowledge of the instrument. And if we have something called weak instrument bias, which I'll come on to later, but essentially that we have a very weak variant that is not particularly strongly related with our exposure, then we could introduce confounding just by the fact that it's not very strongly related to our exposure, so it might, just by chance, be related to something else, which could be a confounder of that relationship. So this isn't particularly, testable in terms of we can't prove it wrong, but we can certainly provide evidence to falsify whether that could be a problem. But obviously it's very difficult to do in a two-sample setting. So if you don't have individual level data, it's very difficult to test whether your exposure and your outcome are independent of your confounding factors, unless, of course, there's been a GWAS of that particular confounder, but then it gets very, very complicated. Then this third one is that we want to make sure that there is no association between our variant and our outcome, independent of the exposure. Now, some epidemiologists in the room may have maybe thought, okay, well, if there's an association between our variant and our outcome, maybe we can just adjust for the exposure of interest to see whether that association goes away, essentially. But we have something in Bristol which we try and advocate against fairly heavily, which is something called collider bias. Don't know, I've seen some nods in the room, so that's quite good. So basically, if we adjust for something on the causal pathway that is essentially a collider between two variables, then we can induce an association between the things that collide on that variable. So in this example, if we condition on the exposure, we potentially could induce an association between our confounders and our genetic variant, and therefore induce an association between our genetic variant and our outcome, which is what we're trying to test. So if we adjust for it, then actually either it could go away, but we don't really know how to interpret that, or we could just induce an association between our genotypes and our outcome. So we try and advocate not to do this, and George is very angry at a group in Japan who have just released a poster that has done this, so he kind of won't talk to them. But anyway, so if you have, the other thing is that if you also have measurement error in something that you're adjusting for, then that can also induce a residual confounding and an error into that association. So essentially, in a one-sample setting, it is very difficult to test whether there is an independent association between your genetic variant and your outcome. So again, it is kind of untestable in a one-sample setting, but it's falsifiable using methods that have been developed specifically in a two-sample setting, which I'll go on to. So that's kind of just the limitations based on the assumptions, but there are many, many limitations. So first of all, I suppose the main key message is that Mendelian randomization, whilst it's compared to a randomized control trial, is just another study design. So we shouldn't be using it as a kind of provative mechanism of understanding the relationship between an exposure and an outcome. It should be used in the context of other studies because it has a whole plethora of different limitations itself. And if we can try and overcome these, then yes, of course, we can then compare it to a trial that's been conducted well, but it should really be used in the context of other studies. Again, for time, and again, if you see any of these that you're interested in, I can talk about them any time this week. But I'm going to mainly talk about the three that I think are interesting at the moment because there are developments in terms of methods to try and overcome these in the kind of space of MR at the moment. So I've got pleiotropy, weak instrument bias, and low power just highlighted here. So pleiotropy, as I said, is another word for this independent pathway between your genetic variant and your outcome. And essentially what this means is that if you're interested in, say, again, funnily enough, the exposure and the outcome of body mass index and cardiovascular disease, if this is your kind of main interest, association of interest, you have a particular variant in a gene which makes a particular mRNA, goes to a protein, and then somehow along the line edits body mass index somehow. And there can be confounders of this relationship, which is what we're trying to overcome with Mendelian randomization and reverse causation. But there could be pleiotropy, meaning that if a genetic variant is created in this framework that creates a different mRNA and a different protein, and then down the line edits a different phenotype that independently affects your outcome, then this can be a problem. Similarly, if your particular variant is in linkage disequilibrium, so it's genetically correlated with something else that also has an independent pathway between your variant and your outcome, then that can also be a problem. And there's lots of different ways now that have been developed to try and overcome this. So first of all, as I kind of said with the CRP example, people are just using variants that have good biological knowledge of what is functionally happening between your variant and your exposure, because that limits the possibility that there is any independent effect of your variant on your outcome. Now, a couple of other methods recently that have been used and developed in a two-sample setting are things like something called MR-EGR. So MR-EGR, the actual EGR part, is actually from systematic reviews. EGR regression is actually used to detect publication bias in systematic reviews and meta-analyses. So Jack Bowden, so this figure here is an excerpt from Jack Bowden's paper. MR-EGR is used to actually not only detect, but try and account for this horizontal pleiotropy. So I'll describe what this is doing, and I like moving, so I'm going to move. So, this is weird. Okay, so on this axis, we have the relationship between the SNP and the exposure. So this is kind of what I showed with that CRP and triglycerides example, but here now we're using summary level data. So we've got SNP exposure, SNP outcome, and all of these dots are, I think, 15 individual SNPs that we are using as instruments for our exposure. This is the true slope here that is the causal effect of our exposure on our outcome. And you obviously can see if you had individual SNPs, so they say this one and this one, the causal effects that we would get from those would be wildly different. So having more in this case is helping. So in a two-sample setting, one of the most strict causal estimators is something called the inverse variance weighted approach, which is essentially just doing the Wald ratio of each of these individual SNPs and meta-analyzing them, weighting those effects by the inverse of their variance. That's what this inverse variance weighted is doing. And it's the most strict because it is literally assuming that there is no pleiotropy, like what we would normally do in an MR analysis. So if there's no effect of our SNP on our outcome, so this x-axis is 0, then there is no effect of the SNP on the outcome. So that kind of makes sense. There's no pleiotropy. There's no independent pathway between the SNPs and the outcome. What this MR-EGA does is allow this intercept to move. So it's literally testing if there is no effect of the SNP on the exposure, what is the independent effect of the SNP on the outcome? So by doing that, not only can you actually estimate the intercept, which is the pleiotropy in this example, it's allowing that to be taken into account in the causal estimation. So here you can see that the IVW, the inverse variance weighted method, which is very strict, is potentially overestimating the slope because it's a lot steeper than this true slope. But the MR-EGA one is a lot closer to the true estimate because we've allowed us to actually estimate that pleiotropic effect. Does that kind of make sense? I'm seeing nodding, so that's good. So now it's a lot easier with two sample approaches to not only estimate but try and account for pleiotropy in this context. And this is a great paper as well, so this explains all of the statistics and the methods that have gone into all of those different approaches. So that's pleiotropy. Weak instrument bias is kind of interesting. So if you have, it's very plausible that if you don't have that many SNPs that are strongly related to your exposure, you might want to choose them, the ones that are, say, less associated. So essentially lowering that GWAS threshold from 5 times 10 to the minus 8 to maybe 5 times 10 to the minus 5, something like that. So you're increasing the number of SNPs that you can use in your MR analysis. Now, not only is there a possible issue of introducing things like pleiotropy by doing that, you can also induce something called weak instrument bias, which is where your SNPs are unable to explain variation in your exposure. So in this case, we have a true negative causal effect between our exposure and our outcome. So increasing levels of your exposure reduces your outcome variation, which is shown in this here. But when you hold the genotypes fixed, there's a confounded positive association. So within each of those groups, there's a positive, I'll do it that way, there's a positive effect of your exposure on your outcome within each of those genotype groups. And as you become, as the kind of panels go from left to right and down to the bottom, you get less and less likely and able to differentiate levels of your exposure. And actually that true negative association starts to look a lot like the confounded positive association. And in this case, this is exactly what happens in a one sample approach when you have weak instrument bias. Your causal effect estimate becomes a lot more like the confounded observational estimate. One nice thing, though, is that in a two sample approach, weak instrument bias actually goes towards the null. So you can then compare, you know which way that bias is being driven, so you can compare the estimates from a one sample and a two sample approach and see whether they're similar, and you know which way that bias is being driven, so you can maybe then determine why they might not be similar. And ways to address weak instrument bias are quite simple in a way. Is first of all, you can use multiple SNPs that are strongly related to your exposure. You can use two sample approaches, where the bias is towards the null. And you can report things such as something called the first stage F statistic, which is what these numbers at the top of the panels were. Essentially, it's just how strong your instrument is, and usually people argue that this is not the case, but usually people say that if it's above 10, then it's a good, strong instrument. And Steve Burgess has done a couple of papers that have kind of proven, or at least given some theory about why this is the case. And then finally, I just wanted to talk about low power, which I think is a problem in most Mendelian randomization analyses, but has become a lot less of a problem with things like UK Biobank, Kadoorie Biobank, so huge samples that are now available for our use. So as I said before with that CRP example, common genetic traits usually only explain a very small proportion of the variation in your exposure. So you need large sample sizes to actually be able to detect an association or effect between your exposure and your outcome. So two sample approaches allow larger sample sizes because GWASs are usually conducted on huge numbers of people. But what you can do is combine multiple genetic variants together into something like a genetic risk score or an allele score or a polygenic risk score. And there's lots of, if you're interested in doing your own MR analyses, there's actually a lot of power calculators that are available online. This is the one that people tend to use quite a lot. So that's a nice way of being able to sort of detect whether you have enough power. And then just briefly, I wanted to talk about MR-base, which is a kind of a nice recent methodological development in this field. So some of you might have already gone onto it, but this is what the website looks like. So it's available in the website and also an R package. So the website's very default heavy. So you can basically just click lots of buttons and then it gives you a result. But the two sample MR package is a lot more flexible, so you can introduce your own parameters to your different methods and analyses. So just briefly, so as it kind of says on its name, MR-base is a database of various summary level data. So I've realized that this is now out of date because essentially we have information from the GWAS catalog. So that's that amazing illustration I showed earlier of all the different SNPs that we know to be related to various traits. And now I think over 100 GWAS studies or consortia that aren't in the GWAS catalog. Plus actually now what we've done is run a GWAS of every single phenotype in the UK Biobank and put that on there as well. So huge number of SNP trait associations in summary level data format. And we have the GWAS catalog, which is just all of the top hits of all of the GWASs that exist, plus however many genome-wide summary level data. So you can use those for outcomes as well, where you're looking up SNPs wherever they may be in the genome-wide data. So it's a database, but it also allows, as Megan kind of introduced earlier, it allows automated MR analyses. So what it kind of does, and this is kind of bringing it back to that selenium and prostate cancer example that I showed way back at the beginning. So we have a very kind of theoretical example, but essentially what MR-base is doing is we have 3 SNPs, and this is this theoretical example associated with our exposure. We can look those, we know that they're related to our exposure due to a exposure GWAS, and we're essentially looking those up in our outcome genome-wide association study. And actually what one of my colleagues did was try and recapitulate what the SELECT trial showed. So what he did was use, I think, 11 SNPs from the most recent genome-wide association study of selenium levels, and then looked those up in the most recent, only published last year, GWAS of prostate cancer. So this is just a reminder of what the observational and the randomized control trial showed, and what he did was actually scale them to the same level of selenium intake that was achieved by the trial. So again, observationally, there was about a 25% reduced risk with about 114 microgram per liter increase in selenium intake, and not much evidence of an association to support that in the randomized control trial. And what the Mendelian randomization showed was, again, not much evidence to support such a substantial reduced risk of prostate cancer. So just a reminder, the SELECT trial took 7 years, $114 million, and this genuinely took about 10 minutes to do. And this wasn't even using MR-base. So this was just using Mendelian randomization. Annoyingly, the most, or at least until MR-base, the most time-consuming and limiting step was getting the data. So that Selenium GUS and the prostate cancer GUS is not publicly available. So you had to go to the, which is a crime, obviously, but if you go to the author, they can give you the data that you need. The basic improvement that MR-base has now given is access to that data in one place. So that's kind of what MR-base does. And this is that same scatter plot that I showed earlier. So you have the SNP exposure association on the x-axis, SNP outcome association on the y, and this with lots of different methods that you can now do in MR-base and in Mendelian randomization methodology. There's no real evidence of an effect. It's pretty straight. And then this is kind of what I was talking about with the kind of meta-analysis. So each of these points represents the Wald ratio of each of those SNPs individually, which is essentially meta-analyzed at the bottom to give you a IVW and the MR-EGA estimate. And again, very, very small evidence to suggest a causal effect. It's riding on the null. And interestingly, what he showed was the off-target effect. So one thing that we're starting to realize that Mendelian randomization is really useful for is predicting if you have a particular drug target that you're interested in or an intervention that you want to introduce into a population, it can test what those off-target effects may be. So here we're showing the effect of selenium on type 2 diabetes, which was that off-target effect that was shown in the trial that obviously wasn't predicted. So it's a nice way of being able to detect whether your intervention or drug may have any detrimental effects other than the outcome of interest. And then very, very briefly, to put a bit of a personal spin on it, because this has all just been what Mendelian randomization is and how it can really help in drug intervention, hypothesis-driven, hypothesis-free effects, estimate, like derivation. I just wanted to show a little bit about what I'm doing at the moment, and if anyone's interested in it or wants to collaborate or ask questions, then I'd be more than happy to stay about. So I come from a group that looks at the relationship between basically the fact that everything is confounded and correlated in an observational setting. And particularly Nick Timpson is interested not just in cancer, but of the effect of body mass index and obesity on everything that it's related to. So for example, I've done some work in cancer, I've done some work in mortality and how body mass index alters that effect. And obviously in observational epidemiology, there could be lots of other things that could alter that association. So one thing that MR has been really helpful, not only in the kind of literature, but also in my kind of career and work, has been looking at the relationship between body mass index and lots of different things. So one thing that I was honored to be involved in was a working group with the International Agency of Research on Cancer, which is based in France, in Lyon. And what they do pretty much every year is generate a handbook that combines all research from cross-sectional studies, from case control studies, from cohort studies, and for the first time ever, Mendelian randomization studies on the effect of body mass index and other adiposity measures on cancer. And what we showed was actually quite nice evidence to support the causal effect of body mass index on a lot of different cancer subtypes, which Mendelian randomization actually kind of reassuringly was supportive of in most of these scenarios. So, you know, epidemiologists took us seriously. And it led to a lot of other kind of maybe unsubtle marketing strategies across the UK. And for example, Cancer Research UK, one of the biggest kind of cancer funders in the UK, took this to heart and actually tried to market obesity as possibly even the second cause of cancer below smoking. So that was quite nice, I suppose. But then what led me to kind of where I am now is trying to understand what might mediate the effects of body mass index on cancer, because as we probably know, Megan and I were talking about this over breakfast, we're not very good at a population level at altering body mass index at a population level or individually. Diet interventions, bariatric surgery interventions have a relatively short-lived. So if we can't really edit body mass index, then what can we alter? And there are obviously lots of things, this is only a very small list of them, that could be to blame, I suppose. And one thing that I was kind of interested in was the gut microbiome, so where this could kind of sit in the relationship between BMI and cancer. So, and I was kind of interested in knowing that Cancer Research UK had actually identified the gut microbiome as something that they were interested in to kind of follow up in terms of a potential intermediary, not just between BMI but various other things and cancer prevention and treatment. So just for you, those of you who don't know, the gut microbiome, and specifically what I work on is the bacteria within the gut, are all the microorganisms that are in the gut and digestive system to aid digestion, to help us against fighting infection and immune responses, protect us against nasty pathogens, and then create essential metabolites that we can't otherwise create ourselves. So what I'm interested in is understanding the links between the gut microbiome and various health outcomes. So my current fellowship is focused on diabetes, colorectal cancer, inflammatory bowel diseases, and I'm now very much in the writing stage of grants to get more funding to look at this in various subtypes of those outcomes. And what really motivated me was because the fact that there'd been exceptional growth in the number of studies that have focused and actually claimed causality in relationships between the gut microbiome and health in humans. But in my opinion, especially in healthy populations and in large scale, had done this with causal assessment that was convincing. And there's a lot of focus on the gut microbiome with industry, with large funders. So that kind of gave me insight into the fact that this probably would have some kind of impact. But I need, in my opinion, better, and actually the funders have asked for a requirement in just better causal inference in this field, and especially triangulation between different study designs. So as I said, MR is just a study design, but if we move against and compare this against lots of different study designs, then we can maybe infer better causality. So hopefully what I've shown you is what we need in Mendelian randomization, which is genetic variants. Now, there have been a couple of microbiome genome-wide association studies. So this is host genetic variation associated with the gut microbiome, but none of them have been comparable. So they've all measured the microbiome in different ways, they've done the analysis in different ways, they've done the genome-wide analysis in different ways, so they're not really comparable, and they've had no overlap or very little overlap in the hits and the bacteria that they've seen relationships with in a genome-wide framework. So what we wanted to do was actually conduct our own with multiple data sets. So one of these was the Flemish Gut Flora Project, which is a large-scale cross-sectional cohort study that has fecal samples as well in Belgium, and it's about 6,000 individuals actually. It's got extensive phenotyping, things like questionnaire, clinic data, blood samples from which they've got genetics, and importantly they have fecal samples and lots of different questionnaires at the same time. That's the PI there with one of the plates. And from those fecal samples, we're able to profile the microbiome phylogeny using various different methods, but the main one being 16S sequencing, which essentially enables you to detect what particular bacteria are present in one of those samples, pretty much down to the genus level rather than species or strain. So what we did was conduct our own genome-wide association study in this sample. We then had 2 German cohorts that we have essentially meta-analyzed with this work. And we found some nice results. So we, in the kind of first meta-analysis of a genome-wide association study with the gut microbiome, we found 2 hits, so one on chromosome 9, one on chromosome 1, that was associated with different types of bacteria in the gut. So 2 that reached our sort of study-wide significance threshold, and then 13 in total that reached conventional kind of genome-wide significance levels, which is brilliant because then that's enabling us to then do Mendelian randomization analyses of these bacteria on various health outcomes. One thing that I did want to say, though, actually, is that functionally very little is known about these particular variants. So we have not much knowledge about biologically what they do. There was also some heterogeneity between the 3 samples. One, kind of the scale of heterogeneity ranged from being pretty consistent between the 3 studies all the way to being fairly inconsistent between the 3 studies. So this is sort of what we're trying to follow up. But nicely, one of the sort of positive controls for our GWAS was one of the most consistently reported SNPs and bacteria that are reported in the literature, which is a SNP in the MCM6 locus, which essentially is associated with whether or not someone can metabolize milk, and bifidobacterium. So if you're lactase persistent or you're able to tolerate milk after weaning, you have lower levels of bifidobacterium in your gut because you don't need it to help you metabolize milk. So what we did was do a 2-sample, so that's using summary level data, bidirectional Mendelian randomization. So bidirectional meaning in one direction we were interested in the relationship between the gut microbiome and various health outcomes that we'd selected based on literature appearance. Then in the other direction, look at the relationship between those health outcomes and the bacteria, mainly because a lot of the studies that exist at the moment are case control studies, so it's very difficult to know whether the disease is causing variation in the microbiome or vice versa. So these are our results, and I won't really go into them, but what was really interesting for us is that, so what we're doing is we've got host genotype and host outcome, and then a bacteria in the middle, essentially. And what we're trying to do is proxy for that bacteria to test whether that has an effect on your outcome. Now, the top hit here, the relationship between a particular bacteria, the presence of a particular bacteria called Dialister, and Alzheimer's disease, was in one direction. And then the bottom is the effect of the diseases on the bacteria. And it's the same thing, so Alzheimer's disease increasing the presence of that particular bug in the gut. Now, interestingly, the top hit, so we use one SNP in all of these associations of the bug on the outcome. That top hit, the SNP associated with Dialister, is in the SORL1 gene, which has been related to Alzheimer's disease in the literature, which brings me on to the complication and why I'm hoping to get funding in the future, is that we're using, as I said, host genotype, bacteria, host outcome. And actually now what we're seeing is the genetic variation linked to the gut microbiome is pretty much just signaling host environment or host disease. So it's really difficult to know whether the causal effect of the bug on the Alzheimer's disease is because the SNP is causing bug variation, is causing Alzheimer's disease, or it's bug, Alzheimer's disease, bug, or actually just the bacteria is a bystander in the relationship between the SNP and the disease. Which brings me on to just a final point about kind of just reiterating this. So that positive control of the lactase persistence variant and bifidobacterium, when we do a causal analysis of bifidobacterium and various adiposity phenotypes like BMI, waist-hip ratio, that kind of thing, we see that lower bifidobacterium increases things like waist circumference, body mass index, waist-hip ratio, but it's very difficult to know whether it's pleiotropic, essentially. So whether the SNP is having an effect on bifidobacterium and then downstream adiposity phenotypes, or whether it's the direct effect of the variant on adiposity phenotypes, which then could go on to changing bifidobacterium, which then questions whether Mendelian randomization is even the right study design in this kind of context, which is exciting for me because I think it could be helpful. So I'm trying to decide how best to do these types of analysis. So, very long-winded summary of what I've just said. So, causal inference in observational epidemiology is difficult. Genetic variants can be used in MR to approve causal inference, not only to validate but to provide evidence against potential observational associations. And it's been used increasingly to prioritize intervention targets and drug discovery pipelines. And MR-based is a really useful exploratory analysis and hypothesis generating platform. But one thing, I've got the little exclamation mark there. One thing is that MR-based has been curated literally by people like me. So there are errors in it. The data is error prone. So if you, go and have fun with MR-based. It's a brilliant platform, but don't click submit and then publish your findings. The reason I wanted to come here and actually talk about MR and have a very long-winded section about the limitations and the assumptions is to understand what MR is, how to maybe see where things might be going wrong, so that when you do go onto MR-based and have a look at it, if you want to, by all means don't, and then have a look in your own data. But if you want to look at MR-based, then do a kind of exploratory analysis. If you find something, then go back to the original studies and do the analysis yourself to check whether it's okay. You would be unsurprised by the number of papers I've read that have either used it and not cited it, or used it very incorrectly and tried to publish something by literally just clicking submit on the website. So don't do that, basically. So there's a couple of references for anyone who's interested in it. But otherwise, thank you so much for having me here. I'm hoping if you want any questions, then ask away. actually. I might show a sneak peek from something. Oh, that's fine. Basically, yes, you can. So, Debbie Lawler in the University of Bristol is essentially the head of a group that is looking at maternal effects and paternal effects on various offspring outcomes. And it's a really interesting field that's moving quite quickly because we realize that there's a lot more complexity in DOHaD-based questions, especially if the outcome in the offspring is pretty much the same as the exposure in the maternal kind of landscape. So like, if, for example, something that I'll present at CHRD is if you're interested in the question between, say, maternal pre-pregnancy BMI and offspring birth weight or BMI, then the MR question would be, okay, well, I want to use genetic variants related to maternal adiposity and test whether maternal adiposity is related to offspring adiposity. But obviously, offspring will inherit those genetic variants either in one way or the other, transmitted or non-transmitted. And it completely it violates the assumption. And also, again, one of the things that was done early in kind of MR and DOHaD questions was adjusting for offspring genotype in that situation. But obviously, offspring genotype is not just maternal, it's paternal as well. And by adjusting for things like that, you can actually induce associations between the maternal genotype, paternal genotype, paternal phenotype, and then all the way around back to offspring phenotype. So there's lots of methods that Debbie's actually published a really good paper that tries to show this and propose different methods to try and overcome it, but it is certainly something that is being done and can be done in the DOHaD question, yeah. Yeah. Thank you. Thank you very much. Oh, wow. Oh, thank you.